So students, now on an ending note to the cellular respiration, we are going to understand three terms. One is fermentation, second is respiratory quotient, and third one is amphibolic pathway. So first let's take into account what is fermentation. We did discuss that in anaerobic respiration, that is one type of respiration where there would be very less amount of energy generation and there would be deficiency of oxygen. That means there would not be an electron acceptor in the end. That is why the Krebs cycle would not be driven and the electron transport system would not be driven. And what happens, the pyruvic acid which is formed in the process of glycolysis, that gets converted either into alcohol or into lactic acid. If I am talking about alcoholic fermentation, it takes place inside the plants, and lactic acid fermentation, you might feel some sort of pain in your muscles whenever you are doing some heavy exercise. That is due to the formation of lactic acid in your muscles, which is in the absence of oxygen. What we do during workout is that we try to make sure that whatever fat is present inside our body, that burns, and when we have to burn something, we need to give it oxygen so that its breakdown takes place and it is released in the form of carbon dioxide and water in the end, and it gives us energy in the return that we are trying to put. So in case the oxygen deficiency occurs inside the muscles, that you are not giving enough oxygen, it often happens, our breathing rate increases while we are doing some sort of exercise. It is trying to give the more and more amount of oxygen to the muscles. Those muscles would go devoid of the oxygen, and because of that, what would happen, you know that when glycolysis ended, we had two molecules of pyruvate, right? Each molecule of the pyruvate, means there are two molecules of pyruvate, each molecule of pyruvate, it breaks down, okay, and it uses NADH, breaks it to NAD positive, okay. It causes it is loss of hydrogen ions. That means it causes its oxidation, basically it causes its oxidation and gives rise to either lactic acid or it gives rise to ethyl alcohol. Now what happens in alcoholic fermentation is, in this case lactic acid would be directly formed, that a carboxylic acid is formed out of the pyruvic acid. There is a conversion, simple conversion, and there is use of energy, no production of energy takes place over here. In case of alcoholic fermentation, what happens is each molecule of pyruvate would break down into one molecule of ethyl alcohol and one molecule of ethanol, as we call it, and one molecule of CO2. Now how it happens? This is an alcohol that is we are getting in the end. Each pyruvate molecule, it breaks down to, and this is not the direct process. What happens is, consider it that each pyruvate molecule is getting broken down to, in the first step, to an acetaldehyde, okay, and acetaldehyde with release of one carbon dioxide molecule, and this acetaldehyde molecule further oxidizes NADH to NAD positive, okay, and gives rise to another CO2 molecule along with what? No, it does not give rise to CO2 molecule, it gives rise to ethanol. Okay, so this is the entire process of alcoholic fermentation, where the pyruvate molecule is getting converted to acetaldehyde with release of one carbon dioxide, and this acetaldehyde further oxidizes NADH and becomes ethanol. This process is known as alcoholic fermentation, and it takes place only if the driving force of oxygen is not present inside the mitochondria, so ethanol would be formed. You know that the entire alcohol industry runs on this particular fermentation reaction, which is taking place by the agency of yeast, which enables the formation of many types of alcohol. So alcoholic fermentation is basically the breakdown of the substrate, that is the energy giving substrate, but in the absence of oxygen. Whenever oxygen is not present, the molecules of carbon dioxide would be released, but what we would get in the end is two molecules of ethanol and two molecules of carbon dioxide would have been released. That is what we get in the end. Same as in the case of lactic acid, we do not get two molecules of carbon dioxide over there. The pyruvate is converted to lactic acid, and this is observed in muscles of animal cells. Now, next topic that we consider is respiratory quotient. As you can see, respiratory quotient is the volume of CO2 evolved per volume of O2 used. So, you can consider it that if the respiratory substrate is using supposedly 2 molecules of oxygen for giving out 2 molecules of carbon dioxide. Let us take the example of glucose, which is a carbohydrate. So, the first case that we consider is those of carbohydrates. In case of carbohydrates, if there is abundant oxygen, that means we are talking about aerobic respiration over here. The examples, the subsequent that I am going to discuss, they would be about aerobic respiration. Carbohydrate, for once, let's consider glucose. You know, it uses 6 molecules of oxygen for release of 6 molecules of CO2. So, the respiratory quotient that we get over here is 1. In case of carbohydrates, our respiratory quotient is 1. In case of fats, the respiratory quotient in case of fats, you know, whenever we have to say that we need to burn fats in our body, we do a lot of exercise, and that exercise needs lots of oxygen supply, oxygen supply, beg your pardon again. Fats would take more amount of oxygen as compared to what they release in the form of carbon dioxide. So, the respiratory quotient would always be lesser than 1. Okay? It would be 0.9, it could be 0.8, it could be 0.5, but it is not going to be more than 1, or it is not going to be equivalent to 1. In case of organic acids, you know, COOH comes in organic acid. It is already having so much of oxygen. So, in any case, the respiratory quotient for organic acids is greater than 1. It could be 2, it could be 3, it could be 4, but it is going to be greater than 1. Alright, and last one is, let's consider the example of anaerobic respiration. When we talk about anaerobic respiration, RQ happens to be infinite. Why it is infinite? Because it is not going to use any oxygen. So, whatever the amount would be, CO2, as much as it is released in the process of anaerobic respiration, amount of oxygen used is always 0. So, what we are going to have is infinite respiratory quotient in terms of anaerobic respiration, and in these we have discussed that in case of carbohydrates it would be 1, in case of fats it would be less than 1, because fats are very large molecules which have fatty acids and glycerol molecule, and organic acids, because they have already lot of oxygen molecules inside them, they are going to have respiratory quotient more than 1. Now, the last topic, that is the last one, ending one for this respiration, is amphibolic pathway. What is amphibolic pathway? That for the breakdown of every type of respiratory substrate, it has to enter the Krebs cycle, so that ATP generation has to take place. Whenever we have any sort of respiratory substrate, we make sure that it enters the Krebs cycle in the form of acetyl coenzyme A. You remember acetyl coenzyme A was the one which enters the Krebs cycle. It binds with oxaloacetic acid and then forms citric acid. So, we need to make sure that whether we are having any sort of respiratory substrate, it could be carbohydrate, it could be fats, it could be organic acid, or consider it to be a protein. So, all these substrate have to somehow reach the condition of acetyl coenzyme A. They have to become acetyl coenzyme A by processes or the steps which have to take place. This particular arrangement which enables all the substrates to enter the Krebs cycle through a uniform pattern is known as amphibolic pathway. Alright, so let's see how the fats would enter the Krebs cycle. Fats would break down into fatty acids and glycerol under enzymatic activity. This glycerol would enter the process of glycolysis and then enter Krebs cycle by the same process that we study under aerobic respiration, by glycolysis, in which we have pyruvate in the end, and then that pyruvate changes to acetyl coenzyme A, and then that enters Krebs cycle. So, basically we are dealing with entering the Krebs cycle by the agency of acetyl coenzyme A. So, these fatty acids, they become acetyl coenzyme A by breakdown, the glycerol enters the glycolysis pathway and then enter the Krebs cycle. So you see that even the fat, all the particles are going to be utilized in the Krebs cycle, which would give rise to the number of ATPs which are generated by any energy source. Next is carbohydrates. That is the simplest one we study in aerobic respiration. We take the example of glucose. It enters glycolysis, then it enters the Krebs cycle. And last one is proteins. These proteins, they break down to amino acids, again under the action of some specific enzymes. These amino acids then get converted to pyruvic acid. Alright, they get converted to pyruvic acid by the process of deamination, and once pyruvic acid is formed, then it undergoes reduction by the process of pyruvate dehydrogenase, and then it enters the Krebs cycle after becoming acetyl coenzyme A. So we have this amphibolic pathway which is followed by every respiratory substrate in the process of aerobic respiration, and every substrate has to enter the Krebs cycle and give whatever amount of energy is expected out of it. Alright, so these are the all basic principles and mechanisms which take place when a cell is undergoing the process of respiration for the generation of energy. Thank you.